Thank you very much for this very kind introduction. It is a pleasure for me to give you an update on Janssen's effort in the development of an HIV and COVID-19 vaccine. I have a conflict of interest as an employee of Janssen Vaccine and a shareholder of Johnson & Johnson. So within Janssen, we are using the Advac technology to build our vaccines, and we have applied this technology indeed for both our HIV and COVID-19 vaccine candidates. By now, we have extensive clinical experience with this platform. It has been used now in over 100,000 participants in the different programs, and these participants were in different age groups, different geographies, HIV positive adults, etc. So we have really a wide range of experience now. These Ad26-based vaccines are using the adenovirus 26 as a vector. So we have made this virus completely innocent by deleting part of the genome and using that room to put in a transgene to which we want to elicit immunity in the context of a vaccine. And we know that our Ad26-based vaccines are well tolerated in the different populations where we have used them, and that they induce good humoral and cellular immune responses with specific functionalities. We also have invested quite a lot in production using a complementing cell line for the missing piece in the genome that makes the virus by itself replication incompetent. And on this cell line, we have been able to produce high yields of vaccine doses with a small footprint, potentially resulting in lower capex and lower cost of goods. By now, we have one vaccine approved based on this technology. This is our Ebola vaccine that was approved in Europe on July 1st of this year. We use sort of the same strategy across our programs. We really like to see first preclinical efficacy of a vaccine candidate by performing animal studies with the vaccine and then challenge with as close as possible virus to what we try to achieve protection to in the human population. Then we try to bridge this efficacy and the immune correlate associated with it with clinical data obtained in phase 1/2a studies, so clinical immunogenicity data, to sort of predict which regimen would likely give similar levels of protection as we have seen in non-human primates or other appropriate animal models. And only if we have that level of comfort, we proceed to phase 2b/3 studies to get the real clinical efficacy in the human population. This is data from a preclinical study with our HIV vaccine, where you can see that after vaccination with different regimens and subsequent challenge with a HIV virus, that we obtained significant level of protection when we used the combination of Ad26-based vaccine and a envelope trimeric protein. And this was the information that we used to further build our clinical development plan. Because from this study, we could establish that M-specific antibodies and T cells correlated with protection against HIV challenge. And we use these immune parameters to also set the bar for human immunogenicity based on which we would decide to proceed to phase 2b and 3 studies. These criteria are listed here. I will not go into detail. I have presented them in earlier cases. But we tested also in the humans the different regimens that we had tested in the non-human primates, and we had set targets for performance of the vaccine relative for these different immune parameters. And what we observed was that not all regimens met this predefined criteria, and only 2 regimens did, which was again vector followed by vector plus protein. Either Ad26 based or MVA based, and in the end we selected the Ad26 regimen containing regimen because obviously that had operational advantages over the MVA based regimen. We are currently executing 2 efficacy studies with our vaccine regimen, which contains of 2 doses of Ad26 based vaccines and then 2 doses of Ad26 based vaccine plus the envelope protein. We are in a fully enrolled phase 2b study in Southern Africa called Imbokodo, which is being executed in young women, and we hope to get results from that study in the course of next year. And we are currently enrolling the Mosaico study, which is a study in MSM and transgender individuals, doing that across Europe and the Americas, because there's a different HIV clade prevalence there, to also get information on potential protection against both those clades and that transmission route. In the meantime, we have also started working on a vaccine against COVID. I forgot to say on the previous slide that, of course, the COVID pandemic has impacted the timelines of our HIV program, but we are trying to catch up, of course, on those. For our COVID vaccine, we use similar technology. So in this case, we put a piece of DNA encoding for the spike protein of SARS-CoV-2 into the At26 vector. We have worked on multiple designs of this spike protein to get optimal stabilization and expression, leading to optimal immunogenicity and also manufacturability in the context of At26. And in from a large panel of variants, so we had 12 mentioned here, but in reality we had even more. We selected a lead candidate called F26CoV2S. Based on preclinical studies, and I'm only listing here, so again, following the same approach that first we want to see efficacy in preclinical models, we build our clinical program. And the most interesting piece, I would say, or most outstanding piece of preclinical data is shown in this slide, where we immunized non-human primates and challenged them 6 weeks later with SARS-CoV-2. We monitored viremia in the lung and in the nose. And you can see here in the middle, animals that received sham vaccination, that after challenge with SARS-CoV-2, we had clear demonstration of viremia, both in the lung and in the nose. That was lasting for at least 10 days in some animals, and some cleared a little bit sooner, but you can see that there's clearly a positive signal. On the contrary, in the animals that received the active vaccine, no viremia could be demonstrated in the lung, and only 1 out of 6 animals had transient and low viremia in the nose after viral challenge. Based on this data and an extensive additional data package obtained in preclinical studies, we moved into the clinic in a phase 1/2A study where we had 3 different cohorts. Cohort 1, individuals from 18 to 55-year-old, cohort 3, individuals above 65-year-old, and cohort 2, again, the younger group, but this cohort would only start if we had identified the regimen that we would use in phase 3, and cohort 2 would then serve as an extension for safety and immunogenicity data. In this cohort 1 and 3, we tested a lower dose of the vaccine, 5 times 10 to the 10th virus particle, as a double or single vaccination regimen. And we also tested 1 times 10 to the 11th virus particles, again as a double or as a single regimen, and the study was placebo-controlled. Primary objective was safety and reactogenicity in both age groups, and secondary objective was immunogenicity of the vaccine. We started the study in Belgium in July with first sentinel cohort. When that went well, we fully enrolled cohort 1, so the 18 to 55-year-olds, and only when that was done, we started enrolling cohort 3, and we did an interim analysis at day 29 post first vaccination. And for sake of time, I only summarize the data. So we had acceptable safety and tolerability at both vaccine dose levels. There was a trend for higher reactogenicity with higher dose level and younger age. A single shot of At26CoV2S elicited neutralizing antibody responses in more than 90% of participants, and also a very nicely Th1 skewed T-cell response. And the vaccine had similar immunogenicity at both dose levels and in all age groups. So based on this, we proposed to FDA to start a phase 3 study, and the FDA approved to proceed with that phase 3 clinical study to test the efficacy of 1 dose of 5 times 10 to the 10th virus particles of At26CoV2S. And this trial started on September 21st. Here's an overview of the overall clinical program as we have currently ongoing or planned. So the first in human study continues. We are now in the phase of the second dose of participants. We are also doing a study, a phase 1 study in Japan, which is now also fully enrolled. And we have a fully enrolled phase 2 study ongoing in the Netherlands, Germany, and Spain, where we are testing additional dose levels and also different intervals between doses to potentially go to a facilitated regimen or a dose sparing regimen if possible. The phase 3 study that I told you about, this ensemble, is now enrolling in US and soon will expand to South Africa, Brazil, and other Latin American countries. And we are planning for a phase 3 efficacy study where we will test a 2-dose regimen for efficacy, so protective efficacy in target population. As soon as we would get a protection, so an efficacy signal from our phase 3 study, we will apply for emergency use authorization. And in the meantime, we are already producing vaccine at scale should we get the positive signal out of our trials, so that we would have a stockpile in case we would get EUA. In parallel, we will also prepare BLA, so it doesn't stop with the EUA. It must be clear from all this work that there's a tremendous group of people involved. I want to mention specifically the HIV team and all our collaborators, and also specifically the people who are working on the COVID-19 program, with a lot of support from BARDA and from US government. And I would like to thank the Janssen team as well for their tireless efforts already since January. And I would like to thank you for your attention.